So hello and welcome to today's webinar, How to Reduce Sample Size Ethically and Responsibly, demonstrated using nQuery. Today's webinar, the hope is that we can show how sample size determination and power analysis can be used to help you explore the options available to you at the study design process and hopefully use that to guide you towards the correct decision for your trial. That which reduces the sample size to the minimum possible while ensuring you get the maximum efficiency and information from your design and trial. Just before we get started, a few common questions. Firstly, in terms of the slides, they can be downloaded from the webinar software as we go along, but if they're not available right now, they will be sent to you afterwards. We're often asked, is the slide or is the demonstration being recorded? Yes, it's being recorded right now. So if you want that, it'll be sent to you automatically after this webinar is complete. And then finally, in terms of questions you may ask, there's a chat window, a questions tab on the right-hand side that you can use to ask questions. I'll answer as many of them as I can at the end of the webinar, but if I don't get around to answering it today, don't worry, I'll be able to get in contact with you by email after the webinar is complete. So hopefully that gives you an idea of the logistics of the webinar. So let's get into the actual meat of it. So as I said, today's webinar is called How to Reduce Sample Size Ethically and Responsibly. Before we get started, I should introduce myself. My name is Ronan Fitzpatrick. I'm the Head of Statistics here at StatSolves, and I've been the nQuery lead researcher since nQuery Interim 3.0. That's about 5 years ago at this point. I've given various guest talks and workshops at places like the FDA, at JSM and similar, and we'll be presenting some poster work on Anderson-Gill sample size at this year's JSM, as well as being involved in the hosting of an nQuery sponsored event for those who are interested in going to JSM this year. So what we'll be covering in today's webinar? Well, firstly, we'll be covering just briefly how study design and sample size interact at the high level, because I think the major thing that I want you to take away from this webinar is that sample size determination and study design are intrinsically linked. They kind of reflect each other, and indeed that study designs that have the best sample size calculations, the ones that get you the lowest required sample size to be able to answer the questions of interest, are those which have been, thought and pre-thought has been put into how to design that study. So hopefully that'll be clear from this webinar. Then I'll talk about some of the common choices that are available to you at the design stage and their likely effect on sample size, and in general trying to advocate for the kind of things that statisticians would generally be in favor of regardless, but showing that by showing how much lower the sample size would be by choosing, in quotes, the correct decision, you can lower the sample size, lower the cost, and make a study which may be considered infeasible feasible. Then there will be a section on adaptive design and how adaptive design can help you with increased efficiency from your trials, and then finally there'll be conclusions and Q&A. Just in terms of what we'll be covering today, we'll be doing some simple t-test and chi-squared analyses, some ANOVA/ANCOVA type analyses, and we'll be doing an unblinded sample size re-estimation for survival type data. As mentioned, this webinar is being sponsored by nQuery and being presented in nQuery. nQuery obviously is sample size determination software widely used within the pharmaceutical and biotechnology fields. And obviously 91% of organizations which had clinical trials approved by the FDA have a license of nQuery at the moment, including some of the companies and organizations mentioned on this slide. So part 1, study design and sample size. And in this, we're just trying to establish what is study design and how does it interact with sample size determination? Because I suppose usually in nQuery, we just talk about sample size determination as a given part of the study design process. We don't really talk about study design too much, except perhaps in the context of our 5 essential steps for sample size, which is available on our website. I suppose just to ensure that you know we're all on the same page for sample size determination. Sample size determination is the process of finding the appropriate sample size for your trial or study. And appropriate's a bit of a generic term, but basically we have some pre-specified idea of what success would mean for our trial, whether that's statistical significance, whether that's having an estimate with a given precision or accuracy, whether that's ensuring that the cost processes or cost constraints that we have are met. These are all considerations for what success would mean in our trial, and there are corresponding metrics to allow you to have a acceptable chance of that success. So the most common, of course, would be statistical power, which is basically your probability of rejecting the null, i.e. having a statistically significant result, given that the alternative hypothesis is true to some specific value. Let's say the effect size is equal to 0.5. And the idea behind sample size determination is that you're trying to balance these ethical and practical issues. I suppose in terms of having too low sample size, which of course is still a fairly common problem, the biggest issue ethically is that putting people into a study and having known or been able to have known beforehand that your sample size wouldn't be powered sufficiently to be able to get a significant result, given that you should get a significant result, i.e. that your treatment is effective, is obviously unethical, because obviously you're putting people through the rigmarole and the risks of a study without giving them the commensurate chance that that study will be able to make accurate conclusions at the end of it. And from a practical issue, obviously, underpowered trials are more likely to just end up with kind of random results, which are then, basically leaving your study success in the hands of the gods rather than based on, appropriate statistical inference. But of course, on the upper side, we do need to, deal with the fact that we can't have infinite sample size due to the cost constraints usually, and therefore it's kind of a Goldilocks problem. We don't want to have too few or too many. And of course, on the higher side, the same risks that come from putting people into a trial which is underpowered are also true for overpowered. If you're putting people into a trial before you've established that a treatment is effective, which of course is the entire objective of a clinical trial, is to find out whether that's true or not, you're obviously exposing people to risks that they shouldn't be when they could be getting the standard treatment. And for this purpose, it's a standard regulatory requirement. In this wider talk, you can talk about things like type M magnitude errors, type S errors, sign errors, and high on. But I think the main thing to take away from this is that many studies continue to have insufficient sample size, particularly in preclinical research. But others are also rejected due to unrealistic sample size needs. Sometimes a statistician does a power analysis for a study participant. They say you need this many sample size. That person turns around and says, I only have this many people. And the problem is that oftentimes that can be due to the decisions that person has made in terms of the design, the person who's asking for the sample size, and they haven't really thought about the effect it has on the sample size. It hasn't really thought on how feasible it makes their study, and it also means that there's a higher chance that they may do sample size determinations or ask for sample size from someone else, or change some of the parameters, say increase the effect size. That kind of thing, which, there's a big thing, of course, people like Steve and Julius talk about when people have the effect size mirror the sample size rather than obviously the effect size being based on some principled idea of clinical significance. So that's obviously something to keep an eye on. So in terms of, that sample size determination, the kind of high level typical introduction that you would get to it. But I suppose the important thing about the sample size is that sample size determination, like when you get to a table in nQuery or you get to sample size for a 2-sample t-test or something like that, really there's a load of steps that should have been taken before you even got to that step. And that's basically asking all of the important design decisions that have gone into creating your trial in the first place. Like, what questions do you want to answer? Usually, say that your treatment is more effective than the standard treatment. What is the primary outcome? Is it blood pressure? And what type of measure of blood pressure are you going to use? Is it a surrogate endpoint? Is it an estimate? All of these questions obviously are becoming more important as time goes on with the whole estimate framework discussion. The grouping structure, paired, crossover, parallel, etc. And what hypotheses are you going to be using? Are you using a superiority, a non-inferiority, etc., etc.? I suppose the important thing to take away from this is that sample size determination should follow the design. To get the sample size, you really must know what success would mean in your trial. Like, if you don't know whether you're powering for a p-value or you're trying to find an estimate with a given amount of precision, you can't do a sample size calculation. It's very important to know exactly what success would mean in your trial, and of course to pre-specify that to ensure the integrity of your trial is upheld. Like, to get the sample size, you need to know what your study outcomes are going to be. Do you have a primary outcome? Do you have multiple primary outcomes? Are you going to power for secondary outcomes? You need to know this before you can do a sample size calculations, and you need to know what statistical method will be used in your trial, ideally to do a sample size calculation. Now, I will mention, like, sometimes you can just rely on a standard method, say, like, if you're comparing two means, a two-sample t-test power analysis might be okay, but generally, you want to get the most accurate sample size probably, and that is a sample size methodology based principled on the statistical model you're actually going to use. And so the important takeaway here is that sample size is emergent from design, not the other way around. So too often, I'm sure most statisticians would agree, what happens is that people come up, I want sample size, I want the sample size from our design, but then they haven't really thought about where their design came from. It hasn't been considered why it has this type of nesting structure, why you're doing an ANOVA analysis, why you have cluster randomized data. And all of those will have an effect on the sample size. So, you know, even forgetting all the other things we're going to talk about in terms of reducing sample size and the other advantages that come from using sample size principally, sample size calculations should happen after the design has been created. And then what we're going to talk about here is where if you believe there's design decisions that have been made that are poor or less efficient than they could be, sample size can then also be used as a tool to help you debate and show why those decisions are poorly thought out or are the wrong choice for your study. And of course, this just kind of summarizes the same, and there's just a little preview here of one of the concepts we'll be covering called Pittman efficiency when it comes to dichotomization of continuous endpoints. But the main thing to think about here is that, you know, design choices will affect your power. The power thus can be used to compare choices, and thus sample size determination can be used in particular to highlight bad choices. And what do we mean by bad? We just mean ones that make your study have higher sample size than required, makes your study more expensive, less feasible, and less efficient overall for very little actual gain in, you know, statistical understandability. Like, you know, I think one of the takeaways we'll probably have is that often people have this idea of simple, like, outcomes that are easily understood, but what often happens is that the cost of those choices is often much higher than people are perhaps aware of in many cases. So how does good design perhaps lead to lower sample size? Well, as we've said, every design choice will have some effect on the required sample size, from the number of groups we compare to the hypothesis we pick to the statistical model. And what we want are good designs which are efficient. That is to say that they maximize, given the resources that we have, the amount of information we can extract about the question or questions of interest to our study. If we have a treatment for cancer, we want perhaps to know, as efficient as possible, does our proposed treatment, our drug for example, reduce mortality? Does it increase overall survival? Does it increase progression-free survival? And all of these other metrics. We need to know that to be able to get approval from regulatory agencies, but also just ethically speaking as people who want to do good in the world in terms of creating treatments that are used in the real world and lead to real gains for patients. And, you know, good designs, lots of decisions here, like randomization level, like most efficient design typically will be randomization at the subject level, not at the cluster level. That's generally just a rule of thumb. Like crossover designs, if they are appropriate, that is to say that you have some kind of chronic disease, and obviously the regulatory agency agrees to it, could be more efficient than a parallel design. It's certainly worth considering that if you believe the disease target is appropriate. And then stuff like which covariates to increase, which I'll talk about specifically at a later point. But, you know, the covariates that we add can reduce our sample size as well. These are all, you know, just a small sampling of some of the choices you can make. So what we're going to focus on in this case is two specific design choices that I suppose are often made incorrectly, you know, for lack of a better term, or are at least made in a suboptimal way too often to kind of ignore. And I think most statisticians will probably recognize these, but I think hopefully the thing you'll take away from here is that sample size determination, sample size software like nQuery can easily and helpfully illustrate that for you in a kind of compelling way for someone of any expertise level in regards to statistics. So the two choices will be the choice of endpoint, basically how do we choose to measure and analyze the data that we get, and then the impact of covariates on our analysis. And hopefully what we'll show here is the value of using your data as it exists. If you gather data, you should be trying to squeeze as much value out of that data as possible, rather than trying to like shove that data into a Precrucian bed that makes it easier to understand, but at the great expense of actually using the information to its fullest. Ultimately, as statisticians, we want to do statistical inferences that are efficient, that maximize our resources, and choosing to simplify or choosing to acquiesce to simplification only leads to more studies failing to be able to answer the questions that they were made to answer in the first place. And that's obviously a huge issue that we should obviously be trying to prevent. So what's this about the choice of endpoints? So how we choose the right endpoint has been an area that's often been neglected. It's kind of been done typically on an ad hoc basis. Basically, someone's gone, people with blood pressure above this threshold, they're people who did well, and people below it did badly, and then just compared that. That'd be something like a responder analysis. But also just a general idea of how to deal with missing data, how to deal with people who cross over, people who don't stay on their regimen during the clinical trial. And of course, this all feeds into a wider issue, which I'm not going to cover today, but this idea of estimates, which are supposed to take account of all of these factors and ensure that the thing that you estimate, you've explicitly thought about how that maps onto the real world, like the real world evidence that you're gathering in this study. Like, if you ignore people who drop out completely, that could create quite a different estimate than if you choose to do multiple imputation, and all of those things have to be considered within that. But I suppose if we're looking at rule of thumbs, and I often think in terms of statistical communication, the thing you're trying to do is give heuristics, the best heuristics possible, rather than trying to explain it all fully. But in general, the heuristic here should be the closer your endpoint is to the actual thing you measured, the better. So what does that mean? That basically means, like, if I'm measuring blood pressure, which is obviously going to be on a continuous scale, I should analyze it as if it was a continual data. If I get repeated events, if I have something that you could have multiple attacks, say COPD exacerbations, I should treat that as repeated events rather than just measuring, say, the time to the first event. Basically, all of the data you have should be used, and this will also be relevant for the covariate section, but all of the data you have for your endpoint, for your outcome measure of interest, should be used, and your analysis method, your model, and by extension your sample size method, should reflect what the actual data looked like. And why is this? Because basically every time you transform your data unnecessarily. Now, obviously, some transformations are fine. Some transformations, like say log transformations, are sometimes required. That's fine. But if you do a transformation without having considered why you're doing the transformation or the value that it adds, then you are in effect wasting it, wasting some information. Because as you put any information through some type of filter or some type of transformation, you ultimately do end up losing some information, some level of understanding of what this looks like in the actual real world outcome basis. And this choice can have a huge effect on the sample size required for your study. So obviously one example is the inefficient, standard in commas, binary defaults that are often used, stuff like responder analysis, where someone, if they're above a threshold, responds, and if they're below it, they don't. And there's other problems with responders, particularly in regards to regression to the mean and issues around that. But even ignoring the regression to the mean issue, and you really shouldn't ignore why that makes responder analysis quite problematic. Even if you ignore that, even if you have an optimal scenario where you're just splitting something which is actually reflective of what you're doing, you're still losing efficiency and you're still going to have higher end. And we're going to show that explicitly. And so sample size determination, as I said, can help you illustrate the true cost of those unneeded simplifications. And hopefully by doing so, you can convince other people why you want to make the choice that you have. Because people obviously like the idea of, say, yes, no, or time, like they're more familiar with log rank tests than they are with negative binomial models. Or they're more, they had an event, so let's just treat it as a binary event rather than, say, taking the time to event, and all these kind of choices that I've talked about that are on the slide. But The important thing is that if you can show that the sample size is much higher because of the choice being made, even if it's the one that feels more comfortable, more natural, then hopefully you can convince people that actually, yeah, maybe I should be doing the recommended thing and using the data as is rather than trying to force it into a framework that kind of is easier or more trivial to understand. Just because some things are hard doesn't mean they're not worth doing. There's a lot of money and effort at stake in any trial, from preclinical to academic, all the way up to phase 3 clinical trials. Obviously, maybe the money's a bit different, the scales are a bit different, but no matter what study you're doing, lots of effort goes into it, and that effort should be respected. And the way to respect that, if you're going to do a quantitative analysis, is to ensure you're getting as much value as you can out of the data you did choose to gather and analyze. So what we're going to do here is just take a simple example, just simvastatin in acute respiratory distress syndrome. This is from a paper from New England Journal of Medicine. Fairly trivial 2-sample t-test example where they were expecting around a 2.6 difference, basically going from 12.7 in placebo up to 15.3 in the treatment group, in the simvastatin group. And for this, they expected 262 people per group post dropout. They had a 3% dropout that brought them up to 540. And standard deviation of 10.6, target power of 80. Nothing too complicated here. This should be relatively simple. I'll also illustrate briefly how this looks in nQuery, just to ensure everyone knows how the software works. So this is what nQuery looks like here. This is where the sample size calculations occur. Each column is an individual calculation. Each row is a required assumption for the calculation, albeit some rows are occasionally optional. On the right hand side, you can see there's a help card here that basically just summarizes, as you select each row, what the definition is, some suggestions and the acceptable entries. And the rows in white are input rows and the rows in yellow are output rows or solver rows. That is to say, if I fill in everything required except for effect size, it will calculate the effect size based on information that's appropriate for the other constraints that I've given. So let's quickly replicate this example and just show that the sample size of these authors that they chose to do was correct. So we just put in our group 1 and group 2 means. You can see the difference is 2.6 as expected. Then we have our standard deviation of 10.6, giving effect size around 2.245, which would be on Cohen's standardized effect scale, probably a small effect size. And then we have a power of around 80%. And we see we get 262 per group as expected from the statement. There's other features here that we're not going to cover today, but if you are interested, there are plotting features available, such as this default power versus sample size plot. And there are things like the output statement here, which just verbally summarizes the result. And you can get a little report summarizing everything from the context menu by selecting the column and right-clicking. If you're interested in the wider feature set of nQuery, please go to our website. There are lots of tutorials on each of these individual feature sets. So what's today's focus? Well, our today's focus is, we've done a sample size calculation and this has got 262 per group. But let's imagine for a moment that instead of doing a sample size calculation using continuous data, they decided that, either you were a responder or you weren't a responder. Basically, depending how many ventilator-free days you had, you're either doing well or you were doing badly. And to be as fair to them as possible, what we're going to do is we're actually going to pick implicitly what is basically the most efficient point if the actual true means were 12.7 and 15.3 to split these into responders and non-responders. And that would basically be the midpoint between our two means, which is 14. So I'm not going to go into the specifics of why that is, but intuitively you can probably understand why the midpoint between our two means, assuming that they're both normal distributions with equal standard deviations, why that might end up being the point at which the most of the lower mean are in the non-responder and the most of the higher mean are in the responder category. It's kind of intuitive, probably the first choice that you make, and it does end up being the most efficient one. basically which I kind of previewed earlier on here in this Pitman efficiency chart. This is the k equals 0. This is the best point, this is the midpoint, and that's where this efficiency is maximized. But as we're going to see, even though the efficiency is maximized by using the midpoint, we're still going to lose a lot of efficiency versus where we were previously. So what we're going to do is we're going to assume, let's just assume that we had that we were correct in our sample size calculation. We have two z distributions with means equal to 12.7 and 15.3, and anyone who has a value below 14 will be called a non-responder, and anyone with a number of ventilator-free days greater than 14 will be called a responder. Greater than or equal to, in this case, just to split the tie, which does create a slight complication, but we'll ignore that for now. So, what will the new sample size be given this? Well, the first thing we need to do is kind of how many people does that actually imply will be responders or non-responders in each of these groups based on that midpoint split. This is a fairly trivial to calculate way to calculate from a conceptual point of view with those strong assumptions about normal distributions, etc. We just take our kind of normal CDFs and basically take the probability that 12.7 basically the CDF of 12.7 assuming a standard deviation of 10.6 and vice versa for 15.3, 15.3 minus 14, fairly standard normal distribution. But you could also easily illustrate this using simulation as well. And obviously simulation could help you do any cut point and with any distribution assumptions as well for each groups. But to keep things simple, let's just talk about two normal distributions and conceptually pick out that our expected proportion who would have success or be responders in the control group, we about 45%, and the proportion in the treatment group, we about 55%. So given the same significance level and the same power, how much sample size are we going to need now? So we'll go to the two sample chi-squared test, no continuity correction, minimizing the sample size there as well. So let's put 0.05, put in our two proportions of 0.4512 and 0.5488. We get an odds ratio of around 1.5. We put in our power of 80%. And now we have a sample size of 411 per group. Now let's take a look at it. 262 per group if we'd use a t-test using the original data. Choosing to make the most efficient responder analysis we could have, making the most efficient dichotomization of our continuous measure, is 411. And what is that like? What kind of efficiency loss we had there? Well, if we take 262 and we divide that by 411, that's like we're looking at nearly a 30% increase in our sample size. That's a fairly significant whack to be got, just so that you could more easily understand the endpoint. So you could more easily use a binary thing to basically turn people into they either succeeded or they didn't succeed, rather than look at how many actual days were they ventilator free on the respective treatments. Using the data as it was, which was measuring how many times they had a ventilator free day within the month, more value extracted, more efficient, lower sample size, and of course this looks close to the Pitman efficiency that was in the previous graph, which is just 2 divided by pi, and that shouldn't be too surprising. That's conceptually what we expect given the rounding, ignoring the effect of rounding here. Obviously these don't exactly equal this because they've been rounded to the nearest integer. So hopefully the next time that you see someone do a binary classification using a continuous measure, basically taking a continuous measure, turning it into a binary measure without any conceptual reason to do so, without any justification beyond it's easier to understand or someone else did it, or responders, that's something I've heard before, you can show them the cost of what you're doing here. Like if you're looking at 150 extra people in each group, so 300 extra people in total, if each of those people is, costs on average $100 or euros to do, 30,000 euro, that's not insignificant. That's like maybe for like a standard trial. In clinical trials, obviously the expenses for adding new subjects goes way through the roof. And so hopefully in a clinical trial, at least in a confirmatory one, this mistake wouldn't be made. But hopefully this illustrates a simple case where making a bad choice has a dramatic effect on the sample size requirement for your study. And this is just something that should never happen without a very, very strong conceptual justification. So let's move on to another topic, which is kind of similar in terms of use information as it becomes available, don't just ignore it. And that's covariate selection. Now there's kind of a wider issue around covariate selection, but mostly what we're focusing on here is hopefully the value of using covariates if they're available. If you have a covariate and you believe it's prognostic, you should use it. That's basically the rule of thumb I'm trying to get across here. But despite this, covariate selection is a, let's say, controversial process in terms of how it's practically done in many settings. You could look at things like stepwise regression, which most statisticians at this point would agree is a very bad way of selecting covariates in a model. But the other one, which is very common and which generally doesn't make any sense, is selecting your variables based on their baseline significance, their basically imbalance across the treatment factor. So let's say you have two groups like treatment and control. You test, is there significant difference in your covariates versus treatment versus control? And if they are, you add it into the model. And if it isn't, you drop it from the model. That is basically, I'm just going to say wrong. That is a bad way to select covariates. It is a bad way to do modeling. And it's also a way which is happening, you're making decisions based on the data after it's occurred. And that also introduces biases into the process that you could easily avoid. So what should you be doing? Instead of either using things like automated procedures like stepwise regression, or just using the baseline relationship between treatment and whatever your outcome is, or perhaps your baseline values versus treatment. You should just basically, before you do the study, you should be thinking about, here are the things we're going to be measuring. Here are the things we're measuring that are going to be available to us that we know before the study occurs. So we know people's gender, for example. We know people's age. We know things like that. So before we get to the final set of data we're going to use for analysis, this is something that we'll know. So it occurs in time sense before, it exists before the actual outcome is measured. And that of course includes baseline as well, which we'll talk about in the example. So the baseline, because it's being measured before the actual outcome, we can say you could have a causative or other relationship with the outcome, and that's fine. So covariate selection should be really based on, or at least the candidate ones that you're going to include. Maybe you might choose to drop some at some point if you could choose to drop them, some of the set if you don't believe they have any value, like just to get your degree of freedom back. But typically the degree of freedom you get back isn't worth the additional bias that you're introducing by allowing yourself that option, and also just even the small amount of variance you're explaining with that covariate. So really, covariate selection should be part of the study design process, and indeed part of your protocol for your study. And you should be thinking about, what do I think could predict or could be related to or just have a relationship with my outcome of interest. Like let's say your ventilator-free days. What are the things that could influence that? Age, for example, and include them based on that principled clinical perspective. And that should be easy to convince non-statisticians to do, because of course that brings them into the fold. That means that their expertise is being valued, and that means that the debates that you need to have about the study in general, about what you should be doing and choosing to measure in the study, is being done before the study occurs rather than afterwards, when there's nothing you can do about it. Which is obviously a well-known problem in study design and statistics. I suppose once you've decided what covariates to add, there is also the question of what you're going to do with them. And obviously there's many different ways here, but we're going to focus here on the value of using ANCOVA and also similar kind of regression models where you add these as covariates as additional parameters. It's a similar principle in both cases. But the major thing we're taking away here is that ANCOVA, or analysis of covariance, is a useful way, a very useful way of showing the value of covariates and showing the value of them versus not only ANOVA just on the outcome on post, like big post here refers to, post intervention. So a simple analysis, say if you had like 2 groups or 3 groups, would be to do a t-test or a paired t-test, or to do something like just an ANOVA analysis on the post intervention data. But another common thing would also be to take the like if you had a baseline measurement, take that away from the post intervention data and use that as your direct measure, as the difference measure, pre-post difference measure. And ANCOVA can be shown to be better than both of them, albeit the difference between ANCOVA and pre-post will be smaller, but also that ANCOVA is just a more principled way of doing it in randomized interventions, in randomized tests. Like it's more complex when it comes to non-randomized data, just due to the fact that one of the assumptions required for ANCOVA is that there's no relationship between the covariate and the actual treatment intervention. So you don't want the distribution of your covariate to be different in each treatment. And of course, randomization kind of takes care of that, or at least on average will take care of that. And of course, the statistical methods, the ANCOVA model itself is based on the principle of randomization. So it is adjusting implicitly for the fact that there is going to be some imbalance, and that's kind of included in the package. But the main thing you can get from doing ANCOVA sample size is to show the value of including covariates by just showing a simple principle, which is that if you include your covariates in a randomized experiment using ANCOVA, you're going to have a smaller sample size requirement, unless that covariate is literally of no prognostic value whatsoever. So hopefully, one other thing that you'll show is that the important thing for covariates is not the relationship between them and the treatment intervention. Like we shouldn't select or consider covariates important by how much the imbalance in the treatment intervention. Like too many old people are getting the treatment versus the control. That's something you should check because it's one of the assumptions on ANCOVA, but it's not that relationship that you should be using to decide whether a covariate is important, whether it's important to actually have an efficient analysis. That's not what's important, but you want to check that on the negative side. Ironically, one could argue that taking treatments, like selecting only covariates based on the fact that they are related with treatment, is actually the opposite of what you should be doing, because of that intervention, even though we're accounting for that by randomization, if it did exist, would actually be problematic in a non-randomized experiment. So it's quite perverse that that's how some people seem to select them in the case of ANOVA and ANCOVA analysis. The key thing is that it's the relationship between the covariate and the outcome. So let's say between age and ventilator, days free of ventilator, that's the important relationship. That's the one that you want to target, and that's the prognostic factors that you should be looking to add at the protocol stage. So let's just take a very simple example here, which was evaluating Botox for neurogenic detrusor overactivity. So this is actually an interesting example because the power analysis they did, which I'm very going to briefly do, which you could do either by a t-test example, very briefly, because obviously we've just done a 2-sample t-test example. In this case they had a difference of 0.75. We'll just put in these values of 4.85 and 5.6, and a standard deviation of 0.85. Giving about kind of what we consider like a medium-large effect size here, and a power of 80, giving 22 per group. But let's do the same thing, but using ANOVA. This will end up being the same thing. Anyone who's done an introductory statistics course on ANOVA and ANOVA analysis will know the relationship for a single input, for a single covariate, or basically for the treatment effect only, that the F test is simply the T test, like the F statistic is simply the T statistic squared, and therefore they are intrinsically related to each other. So we're going to just use the side table down here to calculate the variance of means that corresponds to what we did there previously, and this is just the same 2 means that we had previously of 5.6 and 4.85. We'll click compute and click transfer. We'll get this variance of means, and the common standard deviation, or within group standard deviation, still has the same definition, giving this smaller effect size here. We have our power of 80 and we get 22 per group. Okay, nothing too difficult here. But what's interesting is that if you go into the actual paper, and this example is on our website, so if you want to see the details, you can get them there. But what's actually interesting is that they had measured baseline outcome for their measure, which in this case was UI episodes or urinary incontinence episodes. So they had measured this at baseline, and they were going to actually use this in an ANCOVA analysis, but they didn't choose to include the effect of that baseline covariate on the sample size. And why they might do that is, well, yeah, it is in the context of a clinical trial. I will say that changes such as adding covariate, which reduce your sample size, are not as encouraged, simply because doing the ANOVA analysis, doing what we just did there, kind of provides what you might consider a ceiling for the potential sample size. Like if the effect of if baseline was completely unrelated to outcome, which to be fair seems unlikely, but if it was, then in that case it wouldn't be too bad, because the ANOVA analysis would still be sufficiently powered in that case. But it is also true that you are probably overpowering in this case. So you have too high a sample size if baseline is a good covariate. And as I said, the focus here is on things that are related to the outcome. We would generally expect that the baseline value that you had before treatment might be related to what your actual paired effect was later on. People who started off high are still high. People who started off low are still lower. Now, obviously, there's a regression to mean effect, so they're kind of mixing up there. But it would be unlikely that someone who started off at the very top of the distribution would suddenly be at the bottom. They might go towards the middle, but they're not going to go right through the middle to the bottom, or at least that's kind of intuitively what we might expect. So even regression to mean, that's only pushing you halfway down. That person's still on average going to be higher than someone who started from the bottom, and so on and so forth. Won't get into the weeds too thick on that, but the important thing here is that they chose to not use the effective covariate. It is conceptually allowable in the context, particularly of clinical trials, to do so, because you have a ceiling sample size now. But for the sake of argument, let's say that we wanted to establish, well, if the baseline was fairly predictive or had varying levels of prediction strength with the actual outcome of strength of UI episodes, what would the effect on sample size be? What would the effect on power be? So in this case, let's just look at the effect on sample size. So what we're going to do is do the same analysis, but on ANCOVA, but let's assume varying degrees of correlation between the baseline measure of UI and the post intervention measure of UI, ranging from 0 to 1 in increments of 0.25. Note, of course, that the correlation coefficient, we're talking about R squared here. We're talking about the percentage of variance explained. That obviously the square root of that is equal to the correlation coefficient rho. Also note that in this case, baseline, I suppose in the baseline case, we expect some violation of the parallel slopes assumption, just because we expect our non-intervention, we expect our intervention effect to not increase the mean in the control and increase it in the treatment. And as you can imagine, you're going to have a positive slope for the treatment group and a non-positive slope for the outcome. And that will when you measure between you expect at baseline both of them to be very close to each other, and then after treatment them to be separated. Obviously that means the slopes aren't parallel, but the ANCOVA method is pretty robust to that violation, and generally the value gained by getting rid of the variance explained by the baseline measurement, which is to say kind of the regression to mean effect to a large extent, that value outweighs, particularly especially in randomized experiments, outweighs the kind of cost that you're introducing there. And obviously there's the obvious cost, which is in any analysis, when you add an additional factor, you are losing a degree of freedom from the residual error term, and that means you are slightly less likely to get sample size. I suppose in extremely low sample sizes, that might be something you might want to consider. But if you have sample sizes that low, I would argue in many cases you really aren't probably going to be powered sufficiently to make any inferences, regardless of whether you do ANOVA versus ANCOVA. But that's just my perspective on that. This side table looks very similar. The only addition is we can have unequal sample size. We transfer that as well. And then the rest of these parameters should look fairly familiar here. We have our number of covariates. We'll set that to 1. And then for the first one, we'll set it to 0. And we'll have our power of 80%. And that gives 42, which is actually, you can see this is like slightly below 80. If we wanted the sample size that was just above 80, this is giving the closest to power rather than always rounding above, then it would be equal to 44. Because of course, you'd have to, assuming we're increasing the sample size in each group at the same time. As in, we don't just increase sample size in group 1, we have to increase in both groups. That'll be easy to show during the power situation where I'll change this to the exact value used here of 44, or because it's total sample size versus sample size per group, obviously. So let's do the same thing now, but instead of setting this having no relationship between baseline and outcome, let's set it that there's 25% in column 2, 50% or 0.5 in column 3, 0.75 or 75% in column 4, and let's just set this to near 1, which would basically imply that our baseline is perfectly predictive of outcome. Now in that case, like conceptually the method is an approximation and ends up giving a result, but obviously you would probably argue practically speaking that if your outcome is perfectly measured by something else, you should include both factors, just because like their perfect correlation obviously leads to the variance inflation, and basically the model goes a bit mad basically in terms of its actual coefficient parameters. So this is mostly a toy example. But the main thing to take away here is that as the correlation between baseline and outcome increases, sample size decreases. And the reason that's happening conceptually is of course that, like every ANOVA model, the test for our effect, so there's going to be like a treatment effect row in our ANOVA table. The F test is for the ratio of the variance explained by our treatment, which is just this value here really, divided by the degrees of freedom, which is just 1 for a 2 sample case, divided by the variance unexplained by the model, with degrees of freedom equal to the remainder not taken up from your other factors in the model. So why is it more efficient to include baseline? Well, if baseline is explaining some of the variation in outcome, or basically explaining some of the variation in our outcome measure, that means that variance is no longer in the residual variance. More of the variation in the total model has been explained by our baseline measurements, which seems obvious, like for obvious reasons. It seems like that would be something that was likely to happen, which means that the residual variance has gone down, and the degrees of freedom has gone down by 1 as well, which does mean, adjusted, it will be slightly bigger for the degrees of freedom effect. But unless your prognostic factor is like really has no relationship whatsoever, that degrees of freedom effect is going to be swamped by the reduction in the variance that has been taken away from the residual variance into the covariate variance term of your ANOVA output, which would usually be like, say, row 2, it'll be something like baseline. And that's why, if you're thinking about your statistical tests, why is it more efficient? Why is your power increasing? That's basically why that's happening in that case. And so in general, and of course, you don't know a priori whether these prognostic factors are going to definitely be good predictors, but I think the takeaway here is that even a relatively low relationship, or squared 0.25, even something as low as say 0.05, isn't going to lead to a higher sample size. So unless you end up being really unlucky and picking 10 prognostic factors, all of which have literally explain nearly 0 of the variance in the outcome measurement, you're generally going to be better off, going to have a more efficient analysis by choosing to having adjust for those prognostic factors, which you based principled things that you think are important from a clinical point of view, or because you thought they might be, reasonable alternative predictors of the outcome, or just because, things that you've measured because you want to measure them for other reasons. Why including them will generally increase the efficiency. So hopefully that illustrates how some simple decisions can help you create better design. Now, I'm going to briefly cover adaptive design as a kind of a more complex approach to achieving the same goal, namely having better sample sizes, more efficient designs, more likely to succeed effectively. But I will note, obviously, adaptive design, it's a much more complex topic, a much wider topic, and this is obviously something which you need experienced people who know a lot about clinical trial design, statisticians, and people who have logistical experience. So obviously the costs here probably go up relative to a non-adaptive design. But I think that the main thing away from this section hopefully is that many of the same principles we've talked about for why those simple things you could be doing, like picking covariates correctly, using the data as it is, these all can a priori be fairly well related to better sample size, lower sample sizes, more efficient designs. And adaptive designs basically use the same principle, because adaptive designs, which are those where you can make any decision or change to a trial while it's still ongoing, such as increasing the sample size, enriching certain subgroups, finding the appropriate dose adaptively, or dropping or adding doses. These all are based, I suppose, on the idea, that principle, that if we knew what the actual efficacy of our treatment was beforehand, we would design the trial in a certain way. Like, we would do that in the most efficient way possible to answer the question and get regulatory approval or whatever other objective we have. Get it to market quickest, basically. And in adaptive design, the idea is that given these choices, based on the interim evidence, based on the evidence as it becomes available, we'll move closer to that, like, for the lack of a better term, that platonic ideal of what that trial could have been beforehand. It's evolving, it's adjusting, it's changing to reflect what we think the truth is, and how can we as quickly and efficiently as possible get to that truth. And the hope then is that you can decrease costs, improve your inferences, and have more successful trials effectively. Having that greater control, that better ability to stack trials together in a seamless fashion. Now, of course, the trial design becomes more complex, the assumptions become more complex, you're going to need to do a lot more simulation, a lot more upfront work to ensure you're not doing something which actually ends up doing the reverse. A poorly designed adaptive design can easily go off the rails and end up costing you more money, and perhaps even having a lower success rate. So, if you had an overly liberal sample size increase regimen, for example, you could end up increasing the sample size way beyond what was needed, and way beyond what perhaps could be justified based on realistic assumptions of what's going to happen. And of course, from a regulatory approval point of view, there's obviously wider requirements there as well. But let's just talk about one example here, just of group sequential design, and then extending that to unblinded sample size re-estimation. Now, I've gone through this example in our last webinar on survival analysis, so I'm not really going to spend too much time going through the detail here. Most of what I'm going to focus on here is looking through the framework of how this reduces sample size. So group sequential designs, which is probably the most common type of adaptive design, is one which facilitates interim analyses. Basically, at pre-specified times, you're going to look at the interim data, see what the effect size is, and decide on a principled way using error spending functions, etc. I won't get into the details here, but using an error spending approach, you can decide, based on the interim evidence, is the evidence so strong at this point that I can stop the trial early because the evidence is very strongly for or against my proposed treatment. That's either stopping for benefit or efficacy or futility. And if neither is found, you just continue to the next look. And the way the sample size is decreased here, because you're spending some of the error, the overall the maximum sample size you require will be higher than for a fixed term trial. But why is the sample size on average going to be lower? Well, if you're stopping early for trials which are, where your drug is just done really well, or uneconomical trials which are unlikely to succeed, you're saving sample size because you're stopping early. And so on average, you can easily end up with a well-designed group sequential design having lower sample size on average over all the potential things that could happen than for a fixed term trial, even if the upfront maximum sample size might be a little bit higher. So what I'm going to do is take a New England Journal of Medicine example here for everolimus, which I've covered in many webinars at this point. And I'm going to just extend it to a tree-look design with an O'Brien-Fleming efficacy bound and a non-binding Huang-Shi-Dekani gamma parameter minus 1.25 futility bound. The details here on this slide. So I'll enter these parameters here. These are just taken from the table on the left on the previous slide. Do note that you need to convert the weeks for accrual and follow-up into months by dividing by 4. I've done the conversion between median survival and exponential parameter here. This is just from the survival parameter converter tool in the assistance menu. So I'll just copy and paste these across. And before we do our calculation, we want to just set up our group sequential design in the side table. On the left-hand side, we have these options that basically set what the group sequential design will be like, how many interim looks will we have, what spending functions will we use, do we want a futility function, what type, non-binding, and we'll just set this to Huang Shi Dakani minus 1.25. And we'll set our sample size ratio to 1 and our power to 92.6, which is what the authors originally had. I imagine in this case they kind of got the sample size first and reverse engineered the power, but let's just say the power was actually 92.6 from the start. And you'll get 191 per group, which is actually higher than the original sample size for a fixed term design, the original design talked about in that paper, whereas around 176 per group. So that's an increase of around 15, right? And then the events have increased from around 282 up to 309. So that's obviously an increase of around 30, well, between 25 and 30. So you've increased the sample size upfront a little bit. But the thing you're going to get back here is that if the interim evidence, if the, say, statistic, the Z statistic at the second look, after 206 events occur, which is less than 282, was greater than 2.511 or less than 1.026, then that would mean that you'll be able to stop early. So that means if your significance level, using nominal alpha here, this is where the significance level is, if your significance level was less than 0.006, or if it was more than 0.152, you would be able to stop the trial early, either for futility if it's above 1.52, or for efficacy if it's less than 0.006. And so that's the values you get here. And if you do simulations, which you certainly should do if you're using any adaptive design or any design at all really, you can get an idea of, well, how likely is that going to lead to, like, what's the average sample size going to be for various outcomes here. But you can even see that under H1, so that H1 here refers to if the hazard ratio was actually 0.666, it's actually telling you here there's a 62% chance or a 70% chance of stopping at either the first look or second look. There's a 62% chance that you will stop at this look based on the fact if your hazard ratio estimate was actually correct. So that's obviously very nice there. And so that's how group sequential design could help you reduce sample size. And then we get into sample size re-estimation. Obviously, we've talked about all the choices you make, including your estimates for stuff like the variance, increase your sample size or decrease your sample size. Obviously, in terms of things like the variance, we don't know them a priori. So it makes sense that what if we just take a sneak peek at them, like the variance or the effect size, based on the interim data, and change our sample size based on that. Note that, I've talked a lot about lowering sample size here, but in the context of sample size re-estimation, I'm generally not encouraged by the regulatory agencies. So in principle, there's nothing really stopping you. So if you're doing preclinical research, these designs like unblinded SSR and blind SSR can actually reduce your total sample size adaptively as well. So there's a lot of background here on unblinded sample size re-estimation, but for our purposes, this is unblinded sample size re-estimation. We've basically said that we're only really going to increase the sample size. So how does that reduce the sample size overall, you may ask? And that's an important question to ask, but basically, with unblinded sample size re-estimation, we only increase the sample size if the effect size is promising. And what that means is that we build in an optionality in terms of when we're designing the trial at the first stages that isn't available for a non-unblinded sample size re-estimation. And what's that optionality? That basically means that we could power for a higher or more optimistic effect size. Let's say that's our expected effect size, like what we expect to happen, but still have the option to increase the sample size and get significant results for lower effect sizes, but ones which are still clinically relevant. So that means, you could put less upfront sample size. You could say, we only need sample size of 200 if our expected result is right, but we can increase sample size and still catch these lower ones, rather than for a fixed term trial, which is where typically what happens is you're supposed to base the effect size on some lower bound for clinical significance. So you're like kind of taking like the lowest possible value that would still be of interest at all to the stakeholders, to the regulator, to the sponsor, etc. And for that reason, the FDA talked about this is a design that could provide efficiency if well designed. But I would say this is a more complex design. Simulation is very important, and it can be less efficient, of course, if you just increase the sample size willy-nilly based on very poor definition of what a promising result is. So the details here in terms of Chendemetz, Cui, Hung and Wang and the different methods and conditional power are provided on our website or in previous webinars. So if you're interested in the details, feel free to do so. But let's just illustrate briefly what this looks like in nQuery. So we're just going to use this interim monitoring and sample size re-estimation button here. This will open up a table like here. This looks very similar to that side table look down here. There's like defining your unblinded sample size re-estimation, and then you're entering your results and basically comparing against these thresholds that you have from this table. So in this case, let's just do the simplest version, which is a Chendemetz and Lan approach, which basically involves constraining when we can increase the sample size. In this case, we're constraining it to 33% based on the absolute lower bound to 92.6%. And we're basically going to say that the hazard ratio, the true interim hazard ratio we end up getting is actually 0.8 rather than 0.6. And if we do that, we see we get a conditional power of around 48%. If we go into, and we'll actually increase the multiplier here just a bit here, and we go to look 2, it's still 0.6. This is obviously a toy example. And 0.8. We add and continue. The n is increased a lot to 850. We go to our final look. We enter our interim hazard ratio of 0.8, and we'll see that we have a significant result. And I'll spoil the end here, which is basically to say that if we were to turn off sample size, which can be done from this menu here, we would quickly see that in this case we would have got a non-significant result. We wouldn't have had significant results in this case, and obviously that would have been a bad thing, because the result here would have still been large enough that we would have been like, that's probably significant. It is above like 1.96, for example. So that's kind of an issue here. But there is an argument here that the sample, like the events increase we've done here, going from 206 events to 850, is very, very significant. So you should really evaluate by simulation whether this is appropriate for your study, whether this has enough efficiency gain to make up for the fact you may end up having up to 900 events required rather than 206. And of course, in the survival study, there's the additional complications that come from the fact that you could either have done this by increasing the sample size to get more events, or just increasing the follow-up. And there is a debate out there that in the survival context specifically, you could have a situation where you could manipulate that to kind of get the result that you want. See the paper by Friedland and Korn in the references for more details. There's also other issues that come with kind of adaptive designs and survival, which are discussed here as well. I discussed these in detail in the last webinar, so if you want more detail, feel free to get in contact and we can share a recording of that. So what are the conclusions? Well, bad design choices like dichotomization, stepwise regression, using pre-post differences, selecting covariates based on relationship to the treatment intervention rather than the outcome. Very common at all levels, sometimes even in clinical trials. And they're poor choices, they're less efficient choices, they're wasting information, they're wasting data. You go to, no piece of data is free. If you're going to collect it, you should use it unless you have strong reasons not to. You have to justify why, you should have to justify why you get rid of information, not the other way around. It shouldn't require a debate about why responder analysis should require justification, but yet oftentimes the debate is in the other direction. Why can't I use a responder analysis? And hopefully sample size determination and power analysis, you can show the cost of those choices. It's easy to explain why 100 extra subjects might be a bad thing, why that might cost more, why that might create more effort and heartache to the people on the ground actually doing the research, and hopefully that can help me communicate why statisticians care so much about these issues and debate so much about why you should do these, and why these heuristics of don't dichotomize, don't use stepwise regression, use ANCOVA are being championed by the relevant researchers. Hopefully software like nQuery or other similar GUI driven software can show easily side by side, like we did with this ANCOVA example, why these different scenarios may make us more likely to pick a certain thing. Show why the value of using data as it is, showing why considering what factors we care about, thinking about what we're going to include in our model a priori is useful and important. And by using nQuery, by using similar software that's GUI driven, you can show that they can look over the shoulder, you're doing it live, and the feedback that I've gotten personally from people over the years is that being able to do that over the shoulder, have that person just see in front of them, the software says why this choice is bad and easily explainable why it is, can be very useful when trying to communicate those results rather than giving like R code or something similar. And adaptive design, in the context of this whole debate of making the best choice, adaptive design, the promise there is that it could give it a chance to move us closer to, I suppose, what would have been the ideal design for what actually was going to happen in our trial. But do consider that adaptive design, it shouldn't be taken trivially. Simulation, good thinking about design, thinking thoroughly about why you're making that adaptation or why you're making that available should be considered carefully. Something like group sequential design, you can probably do with relatively little input. Its merits are well known, but something more innovative like a sample size re-estimation using unblinded data or enrichment, that needs a bit more careful thought and certainly, but the promise is there. It's obviously a lot of research and practical interest in clinical trials and doing them. So it's real nice. It's a good time to get involved in that debate, get involved in thinking about design and thinking about the value that good design adds in terms of creating more efficient designs, lower cost designs, and basically reducing your sample size and getting the most power out of your analysis as possible. So I think we're just about done here in terms of time. Might have run a little bit over. I'll be looking at the questions and questions we've been sent in. I'll be answering a couple of them here. But any questions that you have that I don't answer now, please feel free to send them in now, and I'll be able to answer some of them. I'll answer all of them by email either today or tomorrow. But if you do want to get in contact directly, info@statsolves.com is our email address for asking general questions. And one other thing I just want to mention is that if you want to try any of the features talked about here, whether you're a pre-existing user who wants to use adaptive design features, which are in nQuery Pro, or you haven't used nQuery before, there's now a new online trial available from statsolves.com/trial. If you just sign up with your name and email address, no download, no installation, access to all nQuery features. You can try it in your browser, very convenient, and you can see are these types of options, is nQuery as a tool, or any of the options or tools or add-ons, are there something that you really want to try and have, and you want to have it available to you in your day-to-day work. The references are at the end of this slide, by the way, including reference to a tutorial here on why selecting covariates based on the relationship to outcome is the right way to do things, as well as other ones that cover the wider issues of dichotomania and other issues in drug development, as well as adaptive design questions as well. So I'm just going to briefly take a couple of moments here and then answer a couple of questions. So just a handful of questions here. One was just asking, they want a little more detail on the talk about recurrent events. It's kind of an area that's kind of increasing in interest, of course, and they're just asking, what were they talking about in terms of recurrent events versus time to event or survival type models. And I suppose the thing to think about here is that, what's traditionally happened with things like repeated measures, like say COPD exacerbations or MNS attacks and other types of recurrent events, is that traditionally what's happened is that either people have taken normal approximations, just treat it as like the mean number of events, or they've used a survival model to measure the amount of time to the first event, and then ignored all subsequent events. And of course, that's a classic example where you have this additional information. You know that they had 3 more attacks in the year. They had 1 or 2 more attacks in the year, and you just ignored those additional events after the first one, or you just took the mean of them and ignored the actual, data structure of the data. It's coming from a kind of like a Poisson process or a negative binomial type process. You know, a mean approximation, usually like using something like the square root transformation, for example. It's fine. It'll be better than using the time to event approach probably, but it's definitely not the same as doing a principled modeling approach, which can deal with stuff like over dispersion, for example. As I mentioned, I'll be presenting on the Anderson-Gill model, which is actually an extension of the log rank test for recurrent events at JSM. I'll have a poster in the biopharmaceutical section. So obviously in the last 5 years since I've been involved, we've added loads of tables related to recurrent events, Poisson regression, negative binomial regression, Anderson-Gill obviously upcoming, and various variations on those. So there's no excuse really not to be doing that type of design anymore, using those type of models anymore. The other question I'm just going to point out now is regarding sample size re-estimation in terms of, I kind of alluded to it briefly. I did go through quite quickly, but just like, you're increasing the sample size, how can that be more efficient? Well, I suppose the two levels of that is one, obviously, if the sample size was too low in the first place and you end up having a non-successful result, that's a lot of inefficiency because obviously you've had the cost of an unsuccessful trial that could have been successful. But on the other hand, as I mentioned, like this point here, just that the FDA can provide efficiency. I think that's, can provide efficiency, be careful. But it is true that, that idea of powering up front for an optimistic effect and then having the option to increase sample size for other effects is important. And I think, this kind of comes down to the idea of the MCID, the minimum clinically important difference. That's what we should use, like in traditional sample size determination. The recommendation from people like Stephen Sen, Stephen Julius, and from nQuery, and for us here in nQuery, would be that the effect size that you pick for your sample size determination should be the lowest, the lowest effect size that would still be clinically relevant. And you should do that because then you're basing it on a principled fashion, and you're ensuring that no matter what happens, no matter what happens, the effects or the range of effects, which is your MCID or higher, could be found, have a reasonable chance of being found by your choice of analysis, assuming your assumptions are correct. But of course, because the MCID will often be lower than what you actually expect to happen, the sample size will increase commensurately to reflect that. Perhaps the promising zone design, this unblinded sample size re-estimation design, is a way we could balance that idea because of, I want to use my expected effects. I want to use the effects I expect to happen, but I still want to be able to deal with this MCID idea. This design is one that, if done in a smart way, in an efficient way, in a well understood way, if you know what you're doing, could provide you the option of being able to do both simultaneously in a better way. And hopefully, by basing on the expected difference, if your expected difference ends up being correct, the lower sample size, you'll probably have a lower sample size overall, even accounting for the effect of GST, etc. If that difference in your MCID and your expected difference is relatively large. So you need to think about this a lot more. You need to do more preparatory work. You need to understand the trade-offs for this type of design. But it can provide many of the same advantages of the more simple and obvious choices that you could be making talked about earlier on in this webinar. So that's all I'm going to be able to cover now. I'll answer the rest of the questions so far, and thanks so much for all the people saying thank you for the webinar. As I mentioned, the recording and the slides will be sent to you after this webinar is complete. And so without any further ado, I'd like to thank you so much for attending and say goodbye.